Biochemistry department, as part of our advanced course on gene regulation in eukaryotes, welcomes Dr. Bill Klein. Dr. Klein is a professor and Robert A. Welch Distinguished Chair in Chemistry, and also Chair of the Department of Biochemistry and Molecular Biology at the University of Texas MD Anderson Cancer Center. Dr. Klein is recognized by the research community for his contributions to our knowledge in gene regulation in retinal cells. Today's lecture features gene regulatory networks in retinal development and degeneration. I leave you with Dr. Klein.
I bring you greetings from the Texas Medical Center. This is one of the largest medical centers in the world, the Texas Medical Center in Houston. And there's some 14 different hospitals, 5 academic institutions, and about 100,000 people come to work here every day. MD Anderson, which is this complex here, and it's growing, it's metastasizing every day, probably a little more than that even. Our building is right here. We see about 80,000 patients a year who are, of course, the MD Anderson specializes in cancer. We see 80,000 cancer patients a year. We have extensive clinical research going on, but we also have basic research. And that building I circled is the one of the buildings for basic research. And in that building is my department, Biochemistry and Molecular Biology, our sister department to your department. So today, I'm going to try to give you an overview of the development of the retina, the vertebrate retina. And our interests are in transcription factor networks that control the development of ganglion neurons in the retina. And I'm going to tell you about ganglion neurons. If you don't know, I'm going to tell you more about it as we go along. Now, our major objective, and the major objective of many people in the field, is to build gene regulatory networks with the idea of improving our predictive power. So if we understand in a more comprehensive way how genes are regulated at a systems level, at a network level, we can understand and predict more about how the cell will form and develop and function. And we're also interested in practical applications. So for example, we're interested in generating animal models that might be relevant for clinical application. And in our particular area, we're interested in generating models for optic nerve degeneration. And I'm going to talk a little bit about that towards the end of the lecture. So what I'm going to do for you today is give you a summary of some of the work we've done and others have done in this field with regard to retina development in general and these ganglion cells in particular. And then I'm going to focus in on some of the research that we've done to build gene regulatory networks. And then finally tell you a little bit about some of the animal models that we've generated with practical application, or at least we hope we'll have some practical application. So let's get started. So first of all, we have to start with the eye, because that's what I work on. And this is the human eye. We're mostly interested in the back part of the eye, closest to the brain, the neuroretina. And I'll tell you more about that as we go along. So as you know, the eye has a very complex structure. It's got many different parts. For this lecture, the most important part is the retina. And the retina is part of the central nervous system. So many parts of the eye are formed through different aspects of development as embryo and fetal development proceeds, and they come from different places. And I'll give you a little bit of that as we get to the next slide. But we're interested in the retina, and that generates as part of the central nervous system. The retina is very, very important because it interfaces one of our important sensory mechanisms, light. It interfaces light with visual perception. So it is the tissue that receives the light, translates it into an electric signal, and then sends it on into visual centers in the brain so that we perceive sight. And how this retina forms, the function of the retina, and the pathologies of the retina have been studied now probably for, oh, I'd say 70 or 80 years in terms of modern biology. Okay, so let's start off looking at how the eye itself develops. And so I'll focus in, this is a scanning EM of mouse eye development going through the different times. So I think some of you are familiar with mouse developmental genetics, but these letters E8.5, et cetera, that refers to the time after conceptus, after the egg is fertilized during embryo growth, and the numbers refer to the day. So E8.5 is 8.5 days after the egg's been fertilized in utero. And what you see first at 8.5 is the neural folds. These are these two structures here. The neural folds have folded together, and you see these little optic pits at the anterior-most part. This is a scanning EM, at the anterior-most part of the neural folds. And these optic pits are going to form the structure, the neural structure of the eye, and that neural structure is the retina. And then as we go through time, I've color-coded the presumptive retina and the developing retina in purple, as you can see. And the retina develops in a very complicated way, in a very beautiful way, looking mechanistically by extensive tissue interactions, morphological movements, and cellular differentiation. So the first thing we see at E.9 is the formation of something called the optic vesicle. And the optic vesicle is really a pushing out of the tissue, and that pushing out is called evagination. And the evagination, which you can see in this little purple color here, is making contact with the surface ectoderm, and the surface ectoderm is going to be the presumptive lens. So the presumptive neural retina and the presumptive neural lens touch each other, and they send signals back and forth. And a lot is known about those signals, but we're not going to talk about those today. That's the very early aspects of the formation of the retina. Then at E9.5, you see that the neural retina and the presumptive lens have now gone the other way. They're pushing inward. They're invaginating. And this invagination causes something called the optic cup, very famous tissue that's been looked at for 80 or 90 years in developmental biology. And this tissue, or these two tissues, are in the midst of forming their particular structures, the neural retina and the lens. And at E11.5, you can now see that extensive morphology and differentiation has occurred. The neural retina has formed, and you can see the lens fibers, the cells of the retina, these stromal epithelial cells, stromal and epithelial cells forming to form the lens. And as you go through time, further differentiation occurs, and you're left with the mature retina. Another structure I should point out is this structure here, labeled P. This is another part of the neural fold, and this is going to form the pigmented epithelium, which you can see forming here, and then finally here. And the pigmented epithelium is a structure in the very back of your retina that when the light that comes through that isn't captured by the retina is then absorbed by this pigmented epithelium. And many retinal pathologies are involved with defects in this pigmented epithelium. And how the pigmented epithelium and the neural retina are formed from this neural tissue, that's also an aspect that's fairly well known now. There's a lot known about that, but we're not going to talk about that today. We're going to focus on the development of the retina. And then the other important thing I want you to see in this particular scanning EM slide is the formation of the optic nerve. So this optic nerve forms somewhere between E13.5 and E15.5. I'll show you some more pictures of that as we go along. And this optic nerve, of course, is the crucial structure that feeds back into the brain. So there are two cables, one from each eye, feeds back into the brain and makes contact with various other centers in the brain, and ultimately goes back to the visual cortex to give us our vision. Again, that optic nerve in terms of pathology is very, very important, and there's lots of diseases that are associated with the degeneration of the optic nerve, which results in blindness and other visual defects. Okay, so that's an overview of eye development. If you take this purple colored structure, this neuroretina that's shown in a particular section in this scanning EM, and turn it 90 degrees, you get a nice image that looks like this. So this is just a simple diagram and confocal micrograph showing you retinogenesis. Retinogenesis is development of the retina. And my whole problem, now you can go to sleep after I say this, because the whole problem is very, very simple. How do we go from this very, very unstructured, diffuse tissue, this neuroblast epithelial layer, to this very beautiful patterned neuroretina that I'll tell you about in panel B, that is able to then perceive light. And in panel C, you see all the cell types and differentiated retina in the mature adult. This is an example from a mouse. So what we've done in panel A and B is we've stained with different stains. The red is just a stain for the nuclei, and that's staining the soma, or the cell bodies of the developing retina, and the green and blue are staining the processes. So what you see in panel A is a neuroblast layer. These are actively dividing neuroblasts that are beginning to send out processes even though they haven't differentiated. And these actively dividing neuroblasts will slowly commit to two different aspects of retinogenesis. One is they'll pattern into these three very nice, what are called nuclear layers. The ganglion cell layer, the inner nuclear layer, and the outer nuclear layer. So patterning is a big deal in the retina. You have to get these progenitor cells, these neuroblast cells, to stop dividing, differentiate into different neural cell types, and then move to different places along this epithelial sheet. And that's what's happening as you go from A to B. And again, we're not going to talk about how the retina is actually layered, laminated in this way. A fair amount's known about that, and we've published on that. But I'm going to focus on the differentiation of the retinal cell types. So each one of these neural layers, or cellular layers, gives rise to specific cell types that differentiate from these progenitor neuroblasts. This outer nuclear layer contains the photoreceptor cells, the rods and the cones. And in between the nuclear layers are what are called plexiform layers. These are where the axons and the dendrites contact each other and make synaptal contacts. And in the very outermost part of the retina, that's actually closest to the brain, here's the pigmented epithelium. Oh, by the way, I should tell you, the retina is put in backwards, so it's not intelligently designed by nature. So the retina is put in such that the light passes through, here's the lens down here, off the picture. The light passes all the way through the retina and is then captured by the rods and cones of the photoreceptor cells and then sent backwards. So the light moves this way, and then it's sent backwards after being transformed to an electric signal back down to the ganglion cell layer. So this top layer are the photoreceptor cells, the rods and the cones. The middle layer, the internuclear layer, are intermediate neurons. And these neurons, which we're not going to talk much about their function or their development, these intermediate neurons receive the signal from the photoreceptor cells. So the photoreceptor cells capture the light, turn it into an electrical signal, and send it to the internuclear layer neurons. And those internuclear layer neurons process the signal in all different ways: color, motion, shades, every way you can think of, depth. So they're processing all these electric signals, and those processing events occur in this outer plexiform layer where the dendrites of the photoreceptor cells make contact with the axons of the internuclear layer. The internuclear layer neurons then, after the processing, do some more processing and send the information down to the ganglion cell layer. And the ganglion cell layer is the final neuron components that process the signal to then go into the brain through the optic nerve. And you can see this stuff here is the optic nerve fiber. You can see it better in the schematic. All this stuff here, they're going to bundle together, form the optic nerve. I'll show you some pictures of that as we go along. So the ganglion cell, yeah?
The animals you're using they don't have color vision, right? Your mouse model, do they have the cones and rods both?
Yeah, so that's a good question. So in humans, we have about 50% of our photoreceptor cells are cones, and about 50% are rods. Cones are the color photoreceptors, rods are the black and white and give us most of the other kinds of information. In a mouse, they actually do have color vision. They can see in color, not so great, but they have about 10 or 15% of their photoreceptor cells are cones. And a lot is known about cone development and cone function through mouse and rat physiology and developmental biology. So they don't have a lot of cones, but they have enough that you can do the studies. There are other animals, so the ratio between cones and rods varies enormously as you go from one mammal to the other, depending on the adaptation of the creature to whatever its environment is. Okay, so this light that's been processed through all this information ultimately gets to the ganglion cell layer. The ganglion cell layer has two cell types. Amacrine cells, which are called displaced amacrine cells, and that's not important, but it's about 50% of the ganglion cell layer. And then the ones that we're really going to emphasize are called the ganglion cells, or ganglion neurons, and those are present down here in the ganglion cell layer. So what we're going to ask is, how do these ganglion cells form, and what are their abilities and what are their functions with respect to developmental features? Okay, everybody with me? All right, so the way these cells form is really quite spectacular, and many people have studied this over the years. They form in waves of differentiation, temporal waves of differentiation. In all vertebrates, not just mammals, but in all vertebrates, ganglion cells are the first cells to form. And one of the reasons we became interested in ganglion cell development is because the ganglion cells were the first to commit to their cell fate. And then you get waves of these other cell types. The first waves really the horizontal cells, the cone cells, and the amacrine cells. And then it's followed by the rods, bipolar cells, and the last cells really are the Mueller glial cells. The glial cells are not neurons, but they derive from the neural retina, and they're the supporting cells of the retina. And you can see the time, and the whole time of retinogenesis actually proceeds through birth, and the mature retina is finally built probably about a week or a week and a half after birth. So the temporal sequence of development of the cell types of the retina is crucial to understanding how the retina develops. And the temporal sequence is really based on lots of mechanistic details, some of which I'm going to talk about, that drive the process. And one of the key things, and one of the reasons I'm giving the lecture in this course, is because one of the things that drives the temporal waves of differentiation is gene expression. And gene expression, of course, is mediated, at least in part, by transcription factors. And so here's a schematic of the different cell types of the retina forming And if you go this way, this is more or less time, and you're looking at the different cell types. And the key thing here is that every cell type has a combination of transcription factors that drives its initial commitment from these progenitor neuroblasts. And in general, though this isn't true for every specific case, in general there's two classes of transcription factors that are involved in the specification event. There are homeobox transcription factors. A very famous one is PAX6, which is eyeless in Drosophila. And we'll talk a little bit about PAX6, but not so much. So homeobox transcription factors are very important for the development of all the different cell types. And you can see PAX6 is used often, and so is another homeobox factor called CRX, is used often for many different kinds of cell types. And then the second class of transcription factors are basic helix transcription factors, and these are referred to as proneural transcription factors, the term coming from Drosophila. And here you can see the names of all these different transcription factors that are involved in forming the different cell types of the retina. The one we're going to concentrate on is a factor called MATH5, which is a proneural BHLH transcription factor. And that's very important for the development of ganglion cells. So we're going to look at the combinations of transcription factors, particularly in ganglion cells, that are involved in the actual differentiation of the ganglion cell itself. All right? Now, how do these different transcription factors come to be expressed in these different cell types? Well, that's a real key question, and nobody really knows the answer to that. And the field is engaged. There's a lot of work going on in trying to understand how the transcription factors get turned on in these progenitor cells to then go ahead and do their jobs, make the specific cell types. And one of the models that's emerged to explain this is called a competence model. And this competence model is really a exchange of external and internal interplay. Extrinsic factors and intrinsic factors talk to each other to push the progenitor cells to different competent states. And that's shown in this little cartoon here. Here again you see the different neural cell types and the glial cell type forming as you go through retinogenesis. And what's been postulated, and there's a fair amount of evidence for this, is that there's each stage of retinogenesis at each time, the progenitor cells are gradually changing their competent state. So the retina itself, the environment of the retina, this external signals, the growth factors, the signaling molecules, are changing over time. And as they change, they configure the progenitor cell in different ways. They change the progenitor cell's genetic program such that they express different transcription factors. These transcription factors then go on and produce specific cell types. And so what we have really is a temporal and spatial model of retinogenesis where we have feedback mechanisms going on between the external environment and the internal environment of the cell. And as you might imagine, as the first cell forms, so here we have a progenitor cell, and it's dividing, and one daughter makes another progenitor cell, the other daughter makes a retinal ganglion cell, or one that's going to commit to a retinal ganglion cell. That retinal ganglion cell can then produce substances because now it's been programmed differently than its mother, than the progenitor cell, and it can send out information into the environment to change the environment of the retina and allow the next cell types to be produced. And so that's the idea, that as you move through retinogenesis, you have this temporal patterning and competence change in the progenitor cells. And I'll tell you, and I may touch back on this as we go along, but more and more it's becoming clear that not only are these cells achieving competence by their environment, but they're also programmed at very early stages to become certain kinds of cells. So there's a fair amount of autonomy in what these cells are going to become, and there's also some plasticity in that they can become multiple cell types at any point during development. All right, so we're going to concentrate on ganglion cells. How do these ganglion cells form? And can we establish a comprehensive way of looking at the transcription factors that actually cause the differentiation of the ganglion cells? So here I'm going to show you a little cartoon of the development of retinal ganglion cells. And here we have the neuroblast layer. These are progenitor cells actively dividing. And these progenitor cells aren't really stem cells, because if you take them out and allow them to grow in culture, they'll eventually differentiate. They're not internally self-renewing. And they're not really stem cells in the fact that they've been programmed to become retinal cells. So these progenitor cells form a competence field. And a competence field has been defined in many different developmental systems as cells that can give rise to a specific cell type, but don't necessarily have to give rise to that specific cell type. They're just competent to give rise to that cell type. And the first competence field that forms in the retina is the competence field that is going to commit to the ganglion cell fate. So one of the interesting things, and I've touched on this just a minute ago, is that the competence field isn't physically localized. It's interspersed all over the retina. So somehow, when these cells form, they have to know that they formed, and yet the cell next to it didn't form, this competent cell. And right now, nobody knows why that happens. Nobody knows how these particular cells get picked. And so that's an interesting question in this field. All right. So once the competent cells form, the next step is the commitment to a ganglion cell fate, and that's shown by these green cells. So not all the competent cells will form green cells, will form committed ganglion cells, but some of them do. And then these cells start to migrate to the ganglion cell layer, the layer closest to the lens, and they begin to differentiate and migrate to their final position in the retina. And then more competent cells form as we go through time. Remember, it's a wave, it's not a spike. And then more cells commit to a fate. My postdoc made this cartoon for me, so I always like to show him. And then more ganglion cells form. So that's how you get the development of the ganglion cells through this process. Now, a very striking image of the formation of ganglion cells is shown here, where we've stained the neuroblast layer just with a nuclear stain, and we've stained the ganglion cell layer with a very early marker. Actually, it's the earliest known marker to ganglion cells, and it's a transcription factor. And the name of this transcription factor is POW4F2, a POW domain transcription factor, and it's also called brain3B. It's a very famous factor. And this factor is reporting the initial differentiation of the ganglion cells. And you can see the ganglion cell layers lit up in green by the formation of these cells. So we want to know, really, how does this all happen? What's going on here? And here's another cartoon to give you just an overview of the key steps, or at least the initial steps, that regulate the specification. I use specification and commitment in the same way, even though there are differences. So here we have the progenitor cells. The red here are the progenitor cells, and two important transcription factors have to be at work here. One is PAX6, which I've already referred to. So PAX6 is a pan-neural transcription factor. It's important for the development of a lot of retinal cell types. And PAX6 is also important for lots of other aspects of the eye. If you take PAX6 away as a germline knockout in a mouse, or you look at a null mutation in a human or in a fly, you don't even get any eye at all. You don't get any lens, you don't get any cornea, you don't get any eye at all. So PAX6 is an important regulator of eye development, If you knock it out specifically in the retina, it takes away lots of retinal cell types. So it's very upstream in a gene regulatory network, very up high in the hierarchy, and it's really called a retinal determination gene in terms of the retina. And you need another factor, which is Notch, which is part of the Notch signal transduction pathway, to be off in cells that are going to give rise or commit to a ganglion cell fate. So the Notch signaling system, I hope that you've studied that, or if you haven't, you're going to. Maybe Jose is going to present that lecture. I don't know. The Notch signaling pathway. But Notch works as a, in this system and in most systems, as a negative regulator. And when Notch is on, cells are actively dividing. Notch is associated with different cancers, with active cell growth, and with some aspects of cell differentiation. But in the retina, Notch is associated with active cell division, so it keeps the progenitor cells dividing, as do many other growth factors. When you take Notch away, that allows the cells to go on and move to a committed state and a differentiated state. And Notch works through transcriptional repressors that are BHLH transcription factors called Hess factors. So Notch has to be off, and PAX6 has to be on. And one of the things I referred to when I showed that cartoon is what causes, PAX6 is always on, it gets turned on very early, but what causes Notch to go off? And a lot of people are trying to find out what causes Notch to go off. So if you're interested in this field and you want to do a good postdoc in a retinal development lab, ask the question, how does Notch go off? And then you'll get famous, which is the goal of all of us, right? Okay. So once the cell commits to this fate early on, you activate MAF5, this pro-neural factor, this BHLH factor MAF5. And when MAF5 goes up, you're gonna have a cell that is able to move on to form a ganglion cell. And one of the first events that happens, at least the way I'm describing it now, is that the brain 3B or the POW4F2, I'll interchange those names, gets turned on. And POW4F2 brain 3B is a differentiation factor that causes the differentiation of the ganglion cells. So MAF5 and brain 3B or POW4F2 are the key transcription factors. They represent very strong nodes in a gene regulatory network. And let me talk about these two factors just briefly. POW4F2 brain 3B is part of a family of transcription factors. And I won't go into all the details of the family, but they're transcriptional activators. They have POW domains and homeodomains, POW-specific domain and POW homeodomain. Brain 3B or POW4F2 is shown here. It binds to a specific promoter element or enhancer element on cis-regulatory elements and activates gene expression. And here in this panel, in this picture from an E13.5 embryo, we've knocked a LAC-Z into the brain 3B locus, and we're asking where is brain 3B being expressed by the LAC-Z reporter. And we can see it's expressed in the dorsal root ganglia along the spinal cord, some parts of the mid and hindbrain, but most importantly, it's expressed in the neural retina. I showed you the antibody staining of brain 3B. So brain 3B in the eye is specifically expressed in the neural retina in the differentiating ganglion cells. So brain 3B is a very important transcription factor for the differentiation of ganglion cells. MAF5 is a member of the mouse atonal family. And as I mentioned a couple of times, it encodes a proneural BHLH transcription factor. Atonal is a Drosophila gene that's required for sensory organ development and for our interest, for eye development. So if you remove atonal from a fly, you get no eye. And as you might imagine, a lot of work's been done on atonal and Drosophila, and many of the features of atonal are related to MAF5. So years ago, Nadine Brown in Tom Glazer's lab at the University of Michigan and Lynn Gahn in my lab at MD Anderson found that the expression of MAF5 coincides very strongly or correlates very strongly with when retinal progenitor cells commit to ganglion cells. So we thought that would be a good gene to knock out and see what the relationship was of MAF5 to ganglion cell development and also to brain3B, PAL4F2. And so here's the two knockouts. So this is now setting the stage for our gene regulatory network by identifying two important transcription factors. And what I'm going to tell you as I march through this lecture is we use two basic techniques. We use mouse developmental genetics, genetically engineered mice, and we use microarray analysis and bioinformatics and all this kind of stuff to try to build a gene regulatory network. So the two techniques that we use really are standard in the developmental field, in the gene expression field, and are essential to building a systems level understanding of development and differentiation. So MAF5 is, as I say here, is essential for retinal ganglion cell competence. It forms the competence field, those orange cells, and for the specification, the commitment step. And you can see that here by a knockout of MAF5. So in a wild-type retina, which is shown here at E14.5, we're reporting with the brain3B LAC-Z. Remember, I told you brain3B is an important transcription factor, but it's also an early marker, the earliest marker for ganglion cells. And you can see that we're looking at the LAC-Z expression, and very clearly in a wild-type mouse, you see at E14.5, brain3B is turned on. When we make a knockout in MAF5, we lose all ganglion cells. They don't form. Brain3B is not there, and if you look, you see the ganglion cells aren't there either. So MAF5 is upstream of brain3B in the fact that it removes ganglion cells very early on in the formation of ganglion cells. Now, PAL4F2 brain3B is downstream. It's epigenetically downstream of MAF5. And we know this because if we look at the same experiment, if we look at brain3B reporting in the knockout retina, here's the wild-type retina, which is basically the same as the one I showed you with MAF5. But if we do a knockout of brain3B, we see that the LAC-Z expression is still there. So the cells can form, ganglion cells can form, and they can migrate to the ganglion cell layer, but they don't differentiate properly. And here's an example of that shown here at E16.5. In the wild-type retina staining, this is a flat mount of the retina, and we're staining with a neurofilament. You can see that the fibers, these are the axons that are emanating from the ganglion cells, they're moving towards the optic disc to bundle together to form the optic nerve. And these green fibers that we're showing with the staining, with the antibody staining to neurofilament, are very healthy, fasciculating, bundling together, and pouring into the optic disc. And you can see in the brain3B knockout, those cells are clear, those axons are clearly abnormal. They don't bundle well. They're misguided, and they don't form a proper optic nerve, and they don't move into the optic disc normally. And this defect, there's several other defects associated with this, causes the ganglion cells to develop abnormally, and eventually they die. So almost all the ganglion cells die by birth in the brain3B PAL4F2 knockout. So PAL4F2 is essential for the normal differentiation of the ganglion cells. It works later than MAF5 in the gene regulatory network. Now, a nice example of the relationship between MAF5 and brain3B is shown in this slide, where we have double immunostaining of a tagged MAF5. Here we've knocked in an epitope tag of MAF5. We don't have a good antibody to MAF5, so what we did is we build a mouse that has an epitope tag MAF5, and that's shown in red, and then PAL4F2 brain3B shown in green. And this is expression at E14.5. Can someone turn the lights down? Lights? Yeah. So you can see it a little better. So what we're looking at here is the MAF5 turning on in the progenitor cell layer, the neuroblast layer, and you see nice red cells forming and or expressing the MAF5. And then in the ganglion cell layer, the differentiating layer, you see the brain 3B PAL4F2 being expressed. So there's a nice relationship between those two genes. And as the red ones commit to ganglion cell fate and begin to turn on brain 3B or PAL4F2, that's this zone here of yellow cells. So this zone is caught in time between MAF5 and brain 3B. So MAF5 turns on brain 3B, and then, very importantly, MAF5 gets shut down. MAF5 turns off. So the cells are now locked in to a differentiated state. They've turned on brain 3B, and they've turned off MAF5. So we call that a lockdown process or terminal differentiation process, where we now know that turning off MAF5 and turning on brain 3B causes irreversibility in that these ganglion cells can't go back anymore. They can't go back and form something else. So this tells us that these two transcription factors have an intimate relationship with each other and are part of a hierarchy, different hierarchical levels of a gene regulatory network. And so to build a gene regulatory network, you need to take a systems approach, and you need to look at a processing diagram. And this processing diagram is shown here, where each one of these little boxes represents essentially a program module that's full of different regulatory factors. We concentrate on transcription factors that are responsible for that module. And these modules can be isolated, sometimes artificially, but sometimes they're separate modules in terms of their functions. And so let's go through these just very quickly, and I'll tell you the ones we're going to concentrate on for the rest of the talk. So the retinal determination factors, I've already told you, are things like PAX6. And the retinal progenitor cell proliferation things are things like Notch and Hess. And these have inputs into the processing unit that's associated with the competence of retinal ganglion cells. And if you're paying attention, you'll know that those are driven by MAF5. And then we move to early differentiation, which is driven by brain 3B, PAL4F2, and I'll show you some other transcription factors. And then we move to later transcription factors, differentiation of the ganglion cells, finally the formation of the optic nerve. Now, we're going to concentrate on this hierarchical processing diagram, but I want to point out to you there's other components that feed in, both by feed-forward and feedback mechanisms that are associated with actually the different networks that drive ganglion cell development. But we're not going to talk about those today. What we're going to focus on are the steps that are associated with competence, differentiation, and of the retinal ganglion cells, and finally the formation of the optic nerve. Now, how do we do these experiments? Well, we turn to a bioinformatics approach, and mostly we've used microarray analysis and other forms of gene expression analysis to identify genes that are upstream, or to identify genes that are dependent on MAF5 for their expression and dependent on brain 3B for their expression, and try to build the network in that way. And the way we do this then is to do microarray analysis, where we isolate embryonic retina from wild-type animals, wild-type retinas, or mutant, either MAF5 mutant or brain 3B mutant retinas, and then do differential gene expression profiling. So we've done a lot of that, and I'm just going to give you some take-home messages. So let's look at the microarray analysis we did with brain 3B first. Okay, so we were talking about how to build a gene regulatory network, and the way we were doing it is through microarray analysis using wild-type and mutant retinas. And this is an example of comparing gene expression profiling between retinas that are making brain 3B and retinas that don't have brain 3B. And I've given you an example of 3 different genes to tell you that that brain 3B regulates some, but all not genes that are dependent on MAT5 regulation. So here's a gene, sonic hedgehog, which is yet another growth factor that's involved in the proliferation of the progenitor cells. And you can see when you take away brain 3B, POW4F2, there's absolutely no expression of sonic hedgehog. Sonic hedgehog goes away. So sonic hedgehog is expressed in the ganglion cells, and it's actually involved in feeding back and keeping progenitor cells at the right number. And so it has interesting properties, and we've published quite a bit on that. But basically, for this lecture, what we want to know is that you need brain 3B to express sonic hedgehog. And you also need brain 3B to express neurofilament light chain, which is a cytoskeletal protein that makes up the cytoskeleton of all neurons. And again, you see when you take brain 3B away, you don't see the neurofilament. But brain 3B doesn't control all the genes that are associated with differentiated ganglion cells. So here's another neurofilament or cytoskeletal protein called internexin alpha. And internexin alpha, no problem. You take away brain 3B and you still see expression. So the take-home lesson here is that and I'm going to talk about this extensively in the next few minutes, is that brain 3B controls some but not all elements of retinal ganglion cell differentiation. And what this predicts then is that there's other transcription factors that are parallel to brain 3B and downstream of MAT5 that are turning on these other genes. So what we're thinking is that there really is a network. It's not just MAT5 to brain 3B to differentiation, but it's MAT5 to a group of transcription factors, one of which is brain 3B, and then to differentiation. And it's even more complicated than that, as I'll show you in a minute. So when we do the same experiment with MAT5, we do gene expression profiling in MAT5 mutant retinas versus wild-type retinas. We see more genes. In the brain 3B, I think we saw 90 genes changing, and in the MAT5, we see about 200 genes changing. And you'd expect that because MAT5 is higher up and it has more drastic effects on the development of the ganglion cells. So in the orange are genes that are downregulated in both brain 3B mutant retinas and MAT5 retinas. And in the black, here's this internexin alpha, are genes that are downregulated in MAT5 but are not downregulated in brain 3B, telling us that brain 3B doesn't do everything, but MAT5 is very upstream and basically takes away all the genes that are associated with retinal development. And you can see, of course, brain 3B is highly dependent on the presence of MAT5 for its expression. And we're particularly interested in all these different transcription factors because that's what helps us to build our network. So we see some of the transcription factors are dependent on brain 3B, the ones in orange, and those we guess would be downstream of brain 3B because their expression goes away when we take away brain 3B. And there's others that aren't affected by brain 3B, by losing brain 3B, but are affected by taking away MAT5. And those we think might be upstream, might be either at the same level of brain 3B in a gene regulatory network or higher up. And I'm going to talk about two of these things. I'm going to talk about something called T-brain2 or eomesodermin, and I'm going to talk about one called islet-1. Okay? And then we also have other functional molecules that are associated with expression, and the expression is affected either by the loss of MAT5, brain 3B, or both. Now, I'm not going to talk very much about this, but this is another component of gene regulation that's very important, and I'll just touch on this. MAT5 also is involved in controlling genes in the progenitor cells, not just in the differentiating ganglion cells. And so MAT5 has another job. Besides specifying the ganglion cells, it also represses proneural genes in the progenitor cells where it's expressed. So if you're expressing MAT5, you have downregulation of MATH3, NeuroD, and neurogenin 2. These are related to MATH5. They're pro-neural BHLH transcription factors. These transcription factors are involved in producing the next cell types in retina development. So when you take away MATH5, you see upregulation of MATH3 and NeuroD, for example. That means that MATH5, when it's present, is keeping those genes at low levels or off, and preventing a MATH5 expressing cell from becoming another cell. So that's part of the whole system that makes up this very complex developmental scheme. But again, I haven't got really time to talk much about that. So this leads us to our first simple gene regulatory network diagram, where we're showing MATH5 at the top of the hierarchy. And MATH5 controls two different branches. One is ganglion cells and one is progenitor cells. And the progenitor cells I'm not going to talk much about, but I just showed you it represses these other proneural genes which are involved in producing other cell types in the retina besides ganglion cells. But what I am going to emphasize is MATH5 is involved in, as I've told you now several times, in the formation of the ganglion cells. And it branches into two pathways. One is the PAL4F2 brain 3B pathway, and one is what we're predicting, a separate pathway, where it turns on transcription factors that are in parallel to the brain 3B transcription factor. And we're going to talk about this one in purple, islet 1, in a minute. Now, brain 3B, it turns on some other transcription factors. So you get another layer in the hierarchy. And one of the genes it turns on is this gene called T-brain2, which is a T-box transcription factor. It's also called eomesodermin. And it turns on these other factors too that I listed. So these are many, many different transcription factors. I've only given you some of them that are associated with this hierarchical gene regulatory network. Now, I won't belabor this, but I'll just show you one or two experiments. T-brain2, or eomes, is expressed in a subset of the cells that are going to give rise to the ganglion cells at E14.5. So what this means, here we have an antibody staining ganglion cells with against the T-brain2. And then in red, you have an antibody against brain 3B. And what we can see here is that E14.5, the early differentiating ganglion cells, the ones that are still migrating to the ganglion cell layer, are not expressing T-brain2. But the later cells are expressing T-brain2. So what this means is T-brain2 is being expressed later than brain 3B. So T-brain2 is downstream of brain 3B. And when I've already showed you, when you take away brain 3B, you take away the expression of the T-brain2. And then I'm not going to go into this in much detail, but here's a wild-type flat mount, just like I showed you before, staining for the axons of the ganglion cells. And here's a T-brain2 mutant, where we've specifically knocked out T-brain2 in the retina. And you can see these fibers in high mag here are not normal. They look the same as, or almost the same as, the brain 3B knockout axons of ganglion cells. But as I say in the title here, the T-brain2, or actually I don't say it, but the T-brain2 gives a milder phenotype because it's not associated with all the events of brain 3B. Brain 3B controls more events than T-brain2 because T-brain2 is downstream of brain 3B. So we have sub-branches, we have bifurcation. As we go down the hierarchy, each one becomes more specialized to do certain functions associated with the differentiation. I won't show you the EM data on this, I'll show you more of that later. And here we've used a bioinformatics approach to identify brain 3B sites in an enhancer element that's associated with the T-brain2 gene. And we've shown that that enhancer element has brain 3B binding sites. And using transgenic mice, looking at retina expression with a LAC-Z reporter, we've established that this enhancer requires brain 3B to turn on expression of T-brain 2, or eomesodermin, the other name for this thing. So what we've demonstrated then is that brain 3B, which is dependent on MAth5 for its expression, it turns on this next transcription factor, T-brain 2, and this transcription factor then turns on other genes. And this is a direct relationship between the T-brain 2 and the brain 3B. Okay, now let's look at genes that might be parallel to brain 3B, be in the same hierarchy as brain 3B. And this is this gene called islet-1, which is a limb domain transcription factor, yet another transcription factor. And here we see, this is not our work, this is a work from Carol Mason's lab at Columbia, where she's shown co-expression, not like the T-brain 2 where you see expression later, but we see that the islet-1 is expressed at the same time as brain 3B, or PAL4F2. And I've already told you, when you take away brain 3B, you still express islet-1. So islet-1 is associated, maybe, we predict, at the same level as brain 3B and can help us build this gene regulatory network. And sure enough, when we knock out islet-1 specifically in the retina, doing conditional knockouts, we see that it displays defects that are very similar to the defects we see when we take away brain 3B. And I won't show you all the stuff. Here's the optic nerve. It's shrunk a little bit, and the cells are screwed up. But basically, here's a marker, the brain 3B or the PAL4F2 marker in wild type, and we see most of the cells are gone in the ganglion cell layer when we knock out the islet-2. So islet-2 is associated much the same way with defects in the ganglion cells when we knock it out. All right. Now comes really the heart of matters in terms of establishing roles of transcription factors in a gene regulatory network. And this is what I want to turn to in the last few minutes to build a network. So in this case, we predict that brain 3B and islet-1 have the same jobs in terms of where they are in the hierarchy, but they're different transcription factors. And we look to see, by doing gene expression profiling, what kinds of genes are dependent on islet-1 and PAL4F2. We see 4 different classes. We see one class where you need both genes to turn on a particular gene, both transcription factors to turn on a particular gene. So here's sonic hedgehog. I talked about that before. And here's PAL4F2. When you have wild type, you see expression. When you have mutant, you don't see expression. You see the same thing for islet-1. So sonic hedgehog gene requires both islet-1 and PAL4F2. So we use the logic and, the Boolean logic and, to explain that pattern. And then we have some that rely on PAL4F2 and not islet-1. And here you see that very clearly. This is a gene called synuclein gamma, which is involved in axon synapting. And you see its expression is not affected when we remove islet-1, but it's drastically affected when we remove PAL4F2. And then we have the other way. We have some genes that are dependent on islet-1, but not on PAL4F2. And so here's this, this is interleukin receptor activated kinase 1. Don't ask me what this is, but it's some gene that's expressed in the ganglion cells. And it's strongly downregulated when we remove islet-1. We didn't do the in situs, but here you can see it's not affected. These are the actual gene microarray data, and you see it's yellow because there's no difference between the wild type and the mutant. This is just a control showing you that in this microarray that this particular gene was downregulated. So in this case, we have an and not logic element. So that's another pattern. And then we have some that aren't controlled by either one, suggesting there's yet other transcription factors that sit in this hierarchical layer. And this is internexin alpha, the one I've talked about before. So it's not changed in either of these particular transcription factors. So what we did is we used a clustering approach. We engaged some computational biologists to help us with clustering, and we looked to see all the genes that were downregulated in MATH5, islet1, and PAL4F2 brain 3B, clustered them all together to see what kinds of clustering we'd get. And this is just a very simple conclusion from this clustering analysis. So here you see MATH5, and red indicates all the genes that are downregulated. And so this is the whole set of MATH5 downregulated genes. And then the interesting thing is PAL4F2 and islet1. Here you see subsets of genes are downregulated. In PAL4F2, here's the cluster of the genes downregulated in the PAL4F2 mutant retinas. And the yellow are all the genes that are not downregulated. These genes, you take away PAL4F2, no problem. They're still expressed. In islet1, you see a different pattern. Here's the genes that are downregulated, and here's the genes that aren't affected. And then right here is the overlap between PAL4F2 and islet1. So we can see the 4 different patterns emerging from this cluster diagram that are associated with the different transcription factors, and that allows us to build a little network that I'll show you in the next slide. And also I want to point out that all the red genes here are transcription factors, so there's lots of transcription factors we can include into this gene regulatory network. I've already told you about this T-brain 2, or EOMES, and that's downregulated by both. And then we have some other transcription factors that are yellow in PAL4F2 and islet1, and those would be further predicted to maybe be doing the same kinds of jobs as islet1 and brain 3B PAL4F2. And we're testing that now. So this gives us the opportunity to create a gene regulatory network. And what I'm going to show you is just one component of the network that we've created. We've published much more extensive networks, but here's one component of the network, just trying to give you an idea of what's happening here. So again, as I've talked about, and this is just basically summarizing things, PAC6 has a positive input into MATH5, and the Notch pathway through the Hess repressors, BHLH repressors, turn off MATH5. MATH5 turns on islet1 and PAL4F2, and we know it also turns on these other transcription factors, TLE1 and MITE1. Now, islet1 can turn on transcription factors like IRAC, that IRAC expression doesn't depend on PAL4F2. It can turn on transcription factors like sonic hedgehog. Its expression depends on both PAL4F2 and islet1. This gene, synuclein gamma, can be turned on by PAL4F2, but it can't be turned on by islet1, etc. So in this way, we build the network, and we show hierarchical levels of transcription factors. The one level I didn't put here is the T-brain 2 EOMES level, which would sit right about here. And you can see how we can build a gene regulatory network. And basically, it's a procedure where we look now at the cis regulatory elements, try to make direct connections. We do microarray analysis, trying to figure out which transcription factors fit into the network. And then when we see those transcription factors, we do knockout experiments to see how they meet our predictions. And the one we're working on now is a gene called MITE1, which is a zinc finger transcription factor that's known in other systems. And we're doing a conditional knockout on MITE1 to see if it fits our prediction. Okay. So the other predictive or computational power of building these kinds of networks is we can now go to our computer biology people, computational biology people, and ask them, how do we fiddle with this system in a computer model? So if we input something and output something, what's the result on the optic nerve? And we're currently trying to develop mathematical models where we can build these kinds of input-output paradigms. Okay, so you might ask, well, you're starting to learn about how a ganglion cell, how a neuron in the retina develops. You know, so what? You know, I want to cure disease, and I want to do this and that. So we're compelled by those kinds of arguments. And in the last few years, last 2 or 3 years, we've started projects to try to look at more applied ways to use this gene regulatory network. And what we started with was one of the central nodes, and the gene that we had spent the most time studying, the brain3B POW4F2 gene. And so what we were asking is, can we apply our knowledge of ganglion cell development to generate an adult mouse model for optic nerve degeneration? So the way we did these experiments was to use yet another genetic engineering approach. And this is a genetic ablation experiment where we take the POW4F2 brain3B locus, this is the gene, and we insert in a cassette. And this cassette has a stop lac Z. These are flock sites. If you don't know what I'm talking about, ask me after the lecture. These are flock sites. And these flock sites will allow, when the brain3B is turned on, will allow lac Z expression. And here you see lac Z expression in the ganglion cells of the retina because it's under the control of brain3B. And then we've got a cassette, or we've created a cassette that has a diphtheria toxin peptide, which is a very potent toxin. One molecule will kill a cell. And that diphtheria toxin is our genetic ablation gene. But it can't work because there's a stop codon between the lac Z and the DTA. So what we do is we cross this. We create a mouse that has this particular brain3B allele. We cross it to a Cre recombinase that has a ubiquitous promoter. And this promoter is driving a Cre gene that's a conditional Cre gene. It's fused to an estrogen receptor that's been modified to respond to tamoxifen, which is an estrogen analog. So when we make mice that have this Cre gene, and put in tamoxifen, we activate Cre. Cre then removes these LOXP sites by recombination, and so it removes the lac Z and activates the DTA and kills the cells. At least that's the theory. I never thought it would work, but we did it and it worked. That's why I'm telling you about it. And so we would lose the expression of the blue color, the reporter gene, in the ganglion cells. So Zhang Zhao and Shuquan Mu in my lab did these experiments, and let me just spend the next few minutes telling you about these results. So the first experiment I want to show you is really the key experiment. What we've done is we've injected tamoxifen into a mouse that's 1 month old and then waited at different times and looked at the optic nerve. Oh, I didn't put that slide in. Okay. To look at the optic nerve. And here's a mouse where we didn't inject any tamoxifen. And what we've done, I hate to show the slide before lunch, but what we've done is we've removed the skull, the skull of the mouse, and we're looking now down into the brain or into the skull of the mouse. And here are the eyeballs of the mouse, and the brain's back here. Pretty gross, huh? For biochemists, I guess. And these structures here are the trigeminal ganglia. And the important structure here is the optic nerve. So here you see the optic nerve is coming, the two optic nerves are coming from the eyes. They're going to the optic chiasm. That's where they cross. And then they go back into the superior colliculus, which is the first center that the optic nerve makes contact with the brain. And I don't show that in this scheme. So this is a control where we don't inject in tamoxifen. This is the mouse that doesn't have the Cre, and this is the mouse that does have the Cre on this side. So here's 1 month after we inject the tamoxifen, And you can see here's the nice optic nerve here in the control. And here's the optic nerve, and these are cross sections of the optic nerve. You see it's a little decreased. And here's 3 months later, and here's the optic nerve of the control. And now you see the optic nerve of the animal where we've activated the diphtheria toxin is degenerated. And you see the structures even, the cross section of the optic nerve is even less. And here's 10 months later. And here you see again the control, and you see the optic nerve is now just basically just a sheath. When we look at the EM, there's hardly any axons in there at all. So we believe this is a very nice animal model for optic nerve degeneration that applies to human optic nerve degeneration, because in humans, the optic nerve doesn't go away in a few days. It degenerates over many, many months. And so we've created now an animal model that can mimic that process by destroying the ganglion cells. Now, another thing that happens in humans as their optic nerve degenerates, and I should tell you that optic nerve degeneration, anytime ganglion cells are damaged, the optic nerve degenerates, because the ganglion cells are the cells that send out the axons that form the optic nerve. Now, the most prominent or most frequent disease of optic nerve degeneration and ganglion cell death is elevated eye pressure in the anterior part of your eye. And that's what gives rise to most forms, open-angle forms of glaucoma. So when there's high pressure in the anterior part of your eye, for some reason that's not really clear yet, the ganglion cells sense that elevated pressure and start to die. They start to undergo apoptosis. Optic nerve degenerates in very much the same way that I've shown you, and you see loss of vision unless you deal with the optic pressure. One of the pathologies that's always associated with optic nerve degeneration and ganglion cell loss is a process called reactive gliosis. And what this means is the supporting cells of the retina, the glial cells of the retina, try to compensate for the loss of the ganglion cells. They sense that something's wrong with the retina, and they start to produce cytokines and other factors to protect the retina. And that takes place in humans all the time, where we show in our animal model that reactive gliosis or increased gliosis occurs in our model. And here's an example looking at glial marker and a marker for gliosis, which I won't go into the details of this, but here's the wild type where we don't have the Cre recombinase, and you can see the red staining is just associated with a certain aspect of glial cells. And here's this MAP kinase, which is associated with some of the glial cells. But the main point here is when we destroy the ganglion cells, using the tamoxifen and activating the Cre, we see an increase in the marker for the glial cells, so the glial cells are becoming more active, and an increase in this kinase, which is also associated with gliosis, which is telling us that even though the retina is still intact, we still see the layers and we still see the cell types except for the ganglion cells, even though it's intact, the retina is responding to the loss of the ganglion cells. So it's mimicking the animal model. Well, when I show these data to people, they always say, well, can these mice see? So we set up an experiment to ask, can these mice see? Here's a very simple experiment where we show that the mice are visually impaired. We take the mice and we put them into a box, actually 2 boxes pasted together, and there's a little door here, and one side is dark and the other side is light. So this is a pretty simple experiment. As a matter of fact, behavioral people would say this, behavioral people say this is not a very good test for sight, but it's good enough for biochemists. And so what we do is we put the mice on the dark side, and we have a light source, so there's a light side of the box, and there's a little door. Now, if a mouse is a wild-type mouse, and here's a DTA mouse, so its ganglion cells are gone. If we put a wild-type mouse on the dark side of the box, it has a tendency to scurry back and forth, but mice don't like the light, and they like to be in the dark. So they'll stay out of the light. So if you can see, if you're a mouse and you can see, you don't want to be in the light. If you're a mouse and you can't see, you of course don't know if it's a light or dark, and you move back and forth with equal frequency. So we put a little detector here, and then we ask, can the mice see or not see? And here's wild-type mice, and you can see that they're avoiding the light side because when they see the light, they scurry back to the dark side. And here's the mice where we destroyed the ganglion cells, and you can see they move back and forth equally. So these mice have impaired vision. All right, so we think we've created a very nice model for optic nerve degeneration. Now what we want to do is cure the mouse, bring back the optic nerves. Well, we haven't done it yet, but let me show you some approaches we've taken to get to that step. So our goal is to use cell transplantation therapy to replace damaged optic nerves. And we're going to use our mouse model to do that experiment. And so the idea is very simple. We're going to inject in some kind of stem cells or progenitor cells from the embryo into this adult that doesn't have the ganglion cells in the optic nerve and see if those stem cells can then colonize the retina and then reproduce the optic nerve. And so I'll show you what we've done with this so far, not a lot, but a little bit. We have two ways of doing this. We're either going to take, we've created mice that have a MATH5 GFP allele, so they express GFP in the MATH5 locus, and so these mice, or stem cells derived from these mice, progenitor cells derived from these mice, will report in green just cells that are MATH5 expressing cells. So we can isolate MATH5 expressing cells. And those are the progenitor cells we want, because I've just shown you in the last hour and a half that those are the cells that are going to give rise to ganglion cells. Those are the top of the hierarchy in our gene regulatory model. So we can either get these from embryos, we can isolate retinas from embryos, and by FACS sorting, purify those cells. We've made some progress on this, but we're just starting. And we can also, and people are now beginning to do this, and so are we can isolate differentiating embryonic stem cells. So embryonic stem cells from the mouse are pluripotent stem cells that can give rise to every stem cell in the mouse. And we have now been doing experiments in collaboration with some Japanese workers, Takahashi and her colleagues at the CDB, the Riken Institute in Kobe. And we've been trying to differentiate embryonic stem cells into MATH5 expressing cells. And here's our initial experiments along those lines. I won't go into the details. If people are interested, I'll be glad to show you. But we go through a whole regime where we put in different growth factor components to get embryonic stem cells to differentiate into retinal progenitor cells that are expressing MATH5. So here's an example of what's called an embryoid body, where we've taken an embryonic stem cell cluster, a colony of embryonic stem cells, and turned it into a mass of differentiating cells by certain procedures. And then we've put in the right growth factors to try to promote the expression of MATH5. And that's shown on these two panels here. This is the low mag and this is the high mag. And you can see this embryoid body is extensively expressing the GFP from the MATH5 locus, the MATH5 GFP expressing. And here's, you can see these cells, and if you look even at higher magnification, I don't have a picture of it, you can see these cells look like neurons. We haven't shown they're neurons yet, but they look like they're starting to become neurons. And so we're, yeah, sure.
That MATH5 GFP construct is under regulation by what promoter?
By MATH5. So we've knocked it in. It's a MATH5 allele. We've knocked it into the MATH5 locus. So what we're interested now is in purifying these MATH5 expressing cells and using them for the stem cell therapy. So that's where we are with our particular experiments. Okay, I think I've gone on as long as I should. And what I'd like to do now is at least give you, if you're interested in all this, some references. First, for a general review of retinal development, Luis Parada at Southwestern has written a very nice recent review on really the molecular biology and the development of the visual system in genes in development. And my colleague Xuxuan Mu and I have written a more focused review on ganglion cell development that just came out in a book chapter. So either of those two reviews are good for you if you're interested in this particular system. And of course, I'm available for questions and discussion, certainly to provide you more information on this. And lastly, to just acknowledge the people who worked in my lab who were associated with this. Xuxuan Mu, who has just taken a faculty position at SUNY in Buffalo, has been involved in all of these different projects. And Yao Fu just recently graduated. We've noticed many graduate students in our graduate school, rather than move on to postdocs, they haven't decided what to do, where to go to postdoc or what to do. So she's staying in my lab for a year while she tries to figure out what to do. And I keep telling her, you got to figure out something because I can't keep paying you. But and Zhang Xiao was the guy who was doing these ablation experiments to make the optic nerve model. Cheyenne Mao is another postdoc in the lab, and Takai Kiyama, another postdoc who are involved in this project. And then, of course, we thank the Eye Institute for helping us, and the Robert Welch Foundation for helping us fund the project. Okay, so thanks a lot for listening.